So hi everybody, I'm Bieneke Bron, and I'm going to talk to you about plague. Yes, the Black Death, the disease that killed nearly a third of the European population in the Middle Ages. It's caused by the bacterium Yersinia pestis, and it was introduced into the United States in the early 1900s. It is currently maintained in the western half of the United States in a rodent-associated cycle, so we're safe here. It's primarily transmitted by fleas, but it's still partly ununderstood how it is actually maintained on the prairie land. Less susceptible rodents, like mice and voles, have been hypothesized to be maintenance hosts for the bacterium, but they've also been hypothesized to be the transporter of infectious fleas during plague outbreaks. And what do I mean by a plague outbreak? That means that highly susceptible rodents, like the prairie dog, have massive die-offs. Why do we care about this? We care about this because prairie dogs are a keystone species of the grassland ecosystem. This means they're crucial for the ecosystem. They shape the landscape, and there's a lot of other animal species dependent on them, among them the endangered black-footed ferret. So to aid the conservation of these endangered and sensitive species, it's important to understand this plague cycle and to manage plague. And a novel plague management tool was developed here at UW-Madison, and that's the oral sulfatic plague vaccine, a vaccine for prairie dogs. And in the summer of 2013, large field trials started to determine the efficacy of this vaccine in prairie dogs. And the natural question that arises is, if we put this bait out on prairie dog colonies, what is the effect on the other small rodents that live out there? So that's my thesis. What is the impact of this vaccine on the non-target small rodents? Do they consume the bait? Do they develop antibodies? Are they potentially protected by it as well? And how does their community competition change? Do we see different rodent species appear on a vaccinated site? Because the study has a really neat design. It has a control site where we distribute a placebo or fake vaccine bait, and a vaccine site where we put out the vaccine, so potentially plague protected population. So we compare these pairs, members of the pairs, over time. And that allows us to study this plague cycle in more detail. And for me, that includes which flea species live on what rodent species, and how does this change between our vaccine and placebo site, and what happens when we actually encounter a plague outbreak. Do we actually see these prairie dog fleas on these small rodents being transported across the prairie, moving a plague outbreak along? Just to summarize, I studied the impact of a prairie dog plague vaccine on the non-target small rodents. And by using this unique system, I try to learn more about this plague cycle in the rodents in the US. Thank you.